Well, thank you. That was a very kind introduction. I feel like you might have taken a few of my punchlines here, too. And thank you so much for joining me for this conversation on such a cold winter's night. So, as you heard, I'm going to be talking about the ocean. And so I thought I'd start with looking at our big, blue, beautiful planet. It's pretty remarkable. And if you think about life on this planet, we're visual people. And probably what you're thinking about are each other, humans. You think about life on the planet, and you might think of whales in the ocean, or elephants on land, or your pet dog. And all of these organisms are actually only a small part of the story, because really we're on a microbial planet. In fact, if you took all of the microbes and put them in a box, filled up that box with no space, and then you took all the animals, so every fish, every whale, every elephant, every human, and put all the animals on the entire planet in a box, which box do you think would be bigger? Right, exactly. In fact, the micro box is almost 50 times bigger. Wow, I know. Isn't that crazy? So we're on a microbial planet. We're also, obviously, on a blue planet. And in fact, that big blue, beautiful ocean makes our planet habitable. So we live on an ocean planet, and I get to study microbes in the ocean. I would call myself a microbial oceanographer. And microbial oceanography is just what it sounds like. It's the study of the abundance, the distribution, and the activities of those microbes in the marine environment. So that microbial planet interacting in the ocean. I really have my dream job. It's taken me on expeditions and to do research all over the world's ocean, every continent and every ocean. We go out and we take samples. We sometimes use airplanes. We sometimes use ships. We sometimes use helicopters. And it means I wear everything from a hard hat to a lab coat to a parka to work some days. I've spent holidays on ships. You can see we decorate sometimes on the portholes and have had really wonderful adventures. And before I talk to you about the rest of the science we're doing, I want to make sure that I acknowledge the team that I work with. And so I want to thank my Dyhrman lab family, recent, past, and present. I couldn't fit all the pictures on here, but these are the individuals who are doing the next generation of ocean science and have contributed to the work that I'm talking about today. I also want to acknowledge the funding that we have from both private foundations and federal agencies that makes this work possible. Like I said, I have my dream job, and I'm really grateful for the science that these organizations support. All right, so let's go back to the ocean. It's big, it's blue. You can look out and again, you might imagine the kind of animals that are there, like whales. Maybe you've gone whale watching. Maybe you interact with the ocean because you really enjoy seafood and you had salmon for dinner or something else. But when I look at that water, I think about the microbes. And in fact, I also think about the chemistry. And in every single drop of water, there are all of these microbes and all of these biomolecules or metabolites that are interacting that form the base of how ocean ecosystems work and how resources are cycled on our planet. In fact, in every 1 milliliter, that's about your pinky size drop of seawater, there are 1 million microbes. These include phytoplankton, which are photosynthetic microbes, and bacteria. And it includes about 10 million viruses. Now don't worry, these are viruses that infect microbes, not people. And in fact, they infect microbes and play a really important role in how the ecosystem works. So in the talk today, I'm going to be showing the photosynthetic phytoplankton in green colors, because just like land plants, they use pigments like chlorophyll to photosynthesize. And I'm going to be showing in red and blue the bacteria that use the organic carbon produced by those phytoplankton as food. Now, in that drop of seawater, there are also about 100,000 different molecules of varying concentrations that act as substrates or food for microbes, that are produced and consumed, and that actually form a chemical language as these microbes are talking to each other. And so if you take this whole gamish of chemicals and microbes and put them together, there are myriad interactions between the microbes and their chemical environment, many of which we're still learning about today. And this vast chemical microbe network, as I said, underpins marine food webs and biogeochemical cycling, or the cycling of carbon, nitrogen, and other elements. So let's just dive in a little bit more. Marine microbes, as I said, are really numerous. So here I have imaged some marine microbes from a very small pinhead-sized drop of seawater that you can see in that cube on my friend Grieg's finger. So what we did is we took a little drop of seawater and we stained all of the microbes in there green or red. And so all of those green and red dots are microbes. Again, in that little tiny drop of seawater. The really faint green dots that might be hard to see are the viruses. Now, if we were to use special microscopes and delve in and look at some of these cells, they might still just look like little blobs. But some of them you can tell apart with a microscope. And marine microbes are functionally diverse. They can do lots of different things. These three are a few of my favorites. So, and we grow them in my laboratory and do experiments on them. So on the left, we have a representative called a coccolithophore because it makes these coccoliths, or those plates, on its cell surface. Those plates are actually chalk. So this cell will mine calcium out of seawater and biomineralize or make chalk plates on its cell surface. You know the White Cliffs of Dover? They're white because of this guy. Those are the chalk plates. The middle one is a diatom. It also has these ornate cell walls, and that cell wall is actually glass. So it will mine silica out of seawater and make these glass cell walls. The last one, don't tell the others, is probably my absolute favorite. It is a kind of cyanobacteria called trichodesmium. And actually what you see there are many cells lined up in filaments, and then those filaments are organized into colonies. We have no idea why it makes those colonies or mechanistically how it does it. But these organisms are special because they can actually use dinitrogen gas that's plentiful in the atmosphere as a nitrogen source, which is a very rare trait that not many microbes can do. One thing that all of these organisms can do is photosynthesize. They're phytoplankton, and so they can take CO2 and light energy and convert it into organic carbon, like sugars, that form the building blocks of the cells and can help them grow, and then are shunted through the rest of the food web as organic carbon. We often will call this POC or DOC. POC just stands for particulate organic carbon, and that means the cell has taken CO2 and turned it into biomass. So again, these are all photosynthetic microbes. Just again, reinforce how important these microbes are in the environment. We can actually visualize them based on their pigment content using satellite imagery. So what I'm going to show you next is a movie put together by NASA where all of the warm colors, the green and the red, is chlorophyll pigments for light harvesting in phytoplankton in the surface ocean. And what you can see is that every place where raw materials, like coming out of the Amazon River there, meets surface seawater, you can get increased amounts of phytoplankton. I never get tired of looking at these movies. It's so amazing. So that is a satellite imaging microbes in the surface ocean. Okay, so these phytoplankton underpin the marine food web, and they draw down and fix, what we call fix, carbon dioxide through photosynthesis. So if we think about what's happening on the globe and we think about what's happening in that drop of seawater, and we were to look at that in a cutaway view of the ocean, it might look something like this, where you have phytoplankton in the surface ocean taking up CO2, producing oxygen, and those phytoplankton get eaten by something bigger and then bigger and bigger, and eventually you get the marine food web. And in fact, those microbes fuel global fisheries, and every shellfish, every fish, every whale, every penguin, every polar bear starts with marine microbes in the surface ocean. So they're critically important to all of those animals that we care about. They're also critically important to moving carbon. So we talked about how they pull down CO2 out of the atmosphere and they produce oxygen for us to breathe. And that then you end up with carbon locked up in cells and exchanged between microbes, all of those different chemical compounds I talked about. And some of that carbon is going to get cycled back and get respired and go back out to CO2. But some of that carbon is going to sink. If it makes it to the deep ocean, it's locked away for about 1,000 years, which is the time it takes for the ocean to circulate. If it makes it to that little arrow and it makes it in a hard part and gets down into the sediments, then that carbon is locked away on geological timescales, millions of years. And this process of moving carbon is critically important to the global carbon cycle and thus to climate. And in fact, if you think about photosynthetic carbon fixation and what's happening on land, we're used to understanding that the Amazon and the Amazon rainforest is important for carbon fixation and biodiversity. But all of those land areas are only half of the global total. The rest happens with microbes in the ocean. So here again, if we look at the amount of green here, which is an indicator called NDVI on land and is chlorophyll in the ocean, you can see that there's green all over the ocean as well as in areas like the Amazon rainforest. And again, this means that there's a lot of carbon moving through the surface ocean. In fact, you can look at this over seasons and see the seasonality both on land and in the ocean. Aren't these animations amazing? I know, microbes in the ocean. All right, so again, if you think about that network, moving that carbon around and making all the fish and all of the organisms that we care about, I just wanted to put one number on it for you, and it's a big one. They're moving about 50 petagrams of carbon per year through that network. A petagram is about 1 billion metric tons. These are really big numbers. So 50 billion metric tons of carbon moving through that network every year. Now, we know that there's a lot of carbon moving in there, but one of the real uncertainties is the fate and flux of that carbon once it gets into the microbes. And this is an area that I study a lot, and it's an area that I think about in terms of what's going to happen in the future. Because if there are changes to how that carbon is moving around, that could have big implications for the food webs and the carbon cycling and climate that we care about. So one of the questions that drives my research is thinking about how resilient is that ocean's chemical microbe network to changing conditions. So some of the questions that we think about that I'm going to talk about today are, what are the rules that govern this interacting network of microbes and chemicals? And what are their sensitivities to a changing ocean? Really thinking about how much carbon is moving through the system from the phytoplankton into the water and into some of those other bacteria. I would argue that this is a pressing time to understand how resilient this network is, because the baseline for studying this network and learning how it operates is already changing. There are, of course, the things that we can see in the ocean, like plastics and garbage and other pollution, but there are many changes happening that we can't see, including the ocean is already getting warmer. So here you see temperature patterns comparing 1950 to 1980 to more present day temperatures. And you can see that almost all areas of the surface ocean are red, meaning they're warmer relative to the past. There's also this graphic that many of us have seen and are aware of CO2 increasing in the atmosphere over time. These are observations taken in the atmosphere in Hawaii. And what happens in the atmosphere happens also in the ocean. That increase in CO2 in the atmosphere is increasing CO2 going into the ocean, and that decreases ocean pH. And what that does is change the chemistry that the organisms that live in these environments are used to. So if we want to go out and understand this network, how do we observe the unseen? We know how African savannas work because we can go and observe what the antelopes are eating and the ibex, and then we can watch the lions and see what they're eating. Microbes, we can't directly observe them that way. And chemicals, of course, we also can't directly observe that way. But it is an exciting time to be doing this kind of work because we have myriad new tools that have really just come online in the last 5 to 10 years to make observations. This includes evolving chemical methods that allow new progress, where we can go in with mass spectrometers and understand what chemicals are in seawater. And from some of this very detailed work, we know that some of the chemicals in seawater act as substrates or food. So you have phytoplankton making food for other bacteria and cycling that carbon around. We also know that there are chemicals like vitamins that help organisms grow and maintain their activities. And we know that there are signals that alter microbial phenotypes. And this is that chemical language that I mentioned in the beginning, where you're basically yelling hello at the microbes. There's also evolving genomic methods that allow new progress. So if you think about a cell interacting with its environment, the blueprint for how that cell works is encoded in its DNA or its genome. And we've been able to capitalize on information and tools and technology and reduce costs that have come from the Human Genome Project and what we call the genomic revolution. So just as we can appreciate that the human genome offered a blueprint for how we work, we're sequencing microbial genomes in the ocean now to ask, how do they work? And again, I like to think about this as having a blueprint for looking forward. And you can envision, as this time cover did, how genomics is helping us understand what our risks are and understand human health. And really what we're trying to do is use those genomes to understand the microbes, what the microbes are capable of, and what their future looks like. Some of the first genomes for microbes came out about 10 years ago, so this has really been a relatively new trajectory. I was on the team that sequenced one of the very first ones, and although the vast majority of microbes are beneficial, this one is a harmful phytoplankton called Oreococcus anaphygephans. It causes brown tides, which are a problem in Long Island Sound embayments. In fact, you might have even seen the brown water in some of these environments. And what you can see in that picture is the normal condition, and then when you get a brown tide. And in fact, we sequenced its genome with support from the Department of Energy and found that it was uniquely adapted to live in human-impacted systems like those Long Island Sound embayments that are high in organic matter and high in metals. Only a few years later, we were able to sequence not one, but multiple strains of genome for the coccolithophore, again, one of my favorites. It's called Emiliania huxleyi, or EHUX for short. You can see the White Cliffs of Dover there. And what we found by sequencing multiple strains is that there's a core set of genes in that genome that all of them have. And then there's some that are unique to different strains. And that unique set, we think, is like a fingerprint of saying, this is the special environment that I grow in relative to another strain. So using these genomes, we now have this opportunity to go out and basically look at this sea of genes and track these populations across diverse environments, including Long Island Sound embayment. This is Great South Bay during a brown tide on the left. And I also do field work in environments like Antarctica, which is shown on the right. That was actually taken on Christmas Day, and we'd got special permission to take a Zodiac to shore, and I could look back and take a picture of the ship that we had been on for about 5 weeks at that point. All right, so in the time remaining here, what I'd like to go through are 3 examples of the different tools that we're using to try to better understand resilience in this chemical microbe network, using some of those tools I just introduced. And basically, we do laboratory experiments to test the sensitivities of the network to different conditions. We do modeling to understand really what we don't know. Can we put the pieces together right, and what are we missing? And then we do lots of field work to go in and try to observe the system and learn the rules for how it's operating. So when we think about laboratory experiments, an example of some of the work that we're doing is that we grow microbes in the lab under different conditions. So this experiment, we were looking at high CO2 relative to a control. And then we go in and we leverage this genomic revolution to basically ask the cells, what are they doing? And we do this not by looking at the genes in the genome, but by actually looking one step further in that cascade at the RNA. So when a cell, if the genome is the blueprint, when you take that blueprint and start building the environment or building the new apartment building, you need to make RNA. That RNA gets turned into proteins. Those are the building blocks of the cell. And eventually, they're going to make the molecules that then are released out into the environment and will be sensed and responded to by other microbes. So it's this biological cascade, and we can query different parts of that cascade to try to ask microbes, who are you and what are you doing? And so for this experiment, we grew cells under high CO2. We also grew them under low nitrogen conditions and low phosphorus conditions. We sequenced all the RNA, so you get this readout that you have to interpret. And then we plotted it up. And here's just an example where the warmer the color, the more RNA you have in a given aspect of metabolism. And there's a whole list of different metabolic pathways on the left. This is just different activities for the cell. And what you can see right away is that each of those columns don't look like the other. They have a unique fingerprint. And this is basically a readout of the activities that are changing between these different conditions. And once we know the sensitivities and what the changing activities look like, then we have the opportunity for better interpreting that in other systems like field populations. So what we are learning from the laboratory experiments are that changes in chemistry influence microbial growth and activities, and we can take that information then and use it, for example, in modeling studies. So the modeling example I wanted to give is really an effort to use a numerical global ocean model to explore how different stressors that we anticipate coming in the future ocean will impact microbial community composition. So we basically put a bunch of microbes in there, put a bunch of the chemistry in there, the physics, and we run that model to see what the microbes do. The three impacts that we were looking at was temperature, so an increased temperature from global warming. We looked at altered nutrient supply, mainly decrease. So you saw how I grew those experimental cultures under low nitrogen and low phosphorus. Those are the kinds of conditions that are going to expand in areas of the ocean. And we looked at increased CO2. So here's some of the experimental data that went into the model. What you see on the left are different growth rate responses to CO2. That's what GRR stands for. So each of the colors is a different group of microbes. Each of the dots is a specific experiment. And anything along the line means there was no change with CO2. They didn't care if the CO2 was higher or not. Anything below means they didn't like it, they didn't grow as well, and anything above the line means they increased their growth with high CO2. And you can see that there's a range of responses. For example, the organism on the right there, which we'll just call PRO for short, because that stands for Prochlorococcus, which is a mouthful, is sitting right on that one line. So Prochlorococcus didn't really care whether or not you changed the CO2. Whereas Synechococcus, another microbe, had a real range of responses, and they were generally positive. So they increased their growth with CO2. So what we're going to look at next is the initial condition of the model run. And you can see that in these maps here. So here's an example with Synechococcus and Prochlorococcus. And we started in the year 2000 to say, here's what their distribution is right now. And you can see that Prochlorococcus dominates in the tropics and subtropics. And Synechococcus is higher latitude and doesn't overlap very much with Prochlorococcus. And then we ran the model on a supercomputer and said, what will this look like, this pattern, in the year 2100 with what we predict for changes in temperature, nutrients, and CO2. And this is what we got. Yeah. Now, it doesn't mean that Prochlorococcus is for sure dying out. This is a model. But it's very striking to see how it disappeared, right? It's very low now. So this was the most abundant organism in that region, is now no longer there, and Synechococcus has taken over. Now, you may think, well, it's one microbe versus another. How different could it really be? But if I were to say, these are both phytoplankton, and if I were to say, hey, if we shifted an ecosystem like the temperate forests with trees as the primary producer to a savanna with grass as the primary producer, we can immediately see that that would have a big impact on the ecosystem and the organisms that live there. And it's the same deal. So if we're shifting these microbial populations that serve as the base of the food web, we need to know what might happen, because this could have a big impact on fisheries. Now, again, this is a model. We don't know that this is going to happen, but it gives us important lessons to think about. We also ran the model where we only changed CO2. That's the output on the right. You can see it looks like the other model where we changed everything. So what this tells us is that in these model runs, CO2 change was the key factor driving this shift in distribution. So the take-home messages from this modeling effort was that CO2 is important relative to other changes that we think are coming. The significance of that CO2 response is relative to competitors. So one of the reasons we think we're seeing that shift between the syn and the pro is that they can compete with each other. And even though Prochlorococcus didn't really care in our laboratory experiments whether CO2 was higher, Synechococcus did better. And so what it's telling us is that these network interactions and competition and interaction between the microbes is important to predicting the outcomes. So the other thing that that means is that we really need to be looking at communities and how communities are responding, not just single organisms in isolation. So really from the models, we learned that interactions, competition, and those networking responses really dictate outcomes. So last, I want to talk about how this really inspired a whole new series of field work where we thought we need to go in and try to trace some of this carbon flow using our observational tools in the field, because we need to be looking at what's going on right now. And this work we did at a long-term oceanographic time series site off of Hawaii. So it's called Station Aloha. This is just a spot in the ocean that the US has been observing about every month for almost 30 years now. And so when you do work in this environment, you can set it into this historical framework of observations. It is located in what we call the North Pacific Subtropical Gyre. It's basically in that box that I'm showing you. It is the largest ecosystem on the planet, covers a large area, and this is nutrient-poor, warm water, where it's a little bit desert-like, so you don't have maybe as many phytoplankton as in other environments, but it covers such a large area, it's critically important to the ocean total. We went out and took observations on a ship, and it was not Columbia's RV Langseth, but I wanted to show this picture because Columbia actually operates one of the ships in the US Oceanographic Research Fleet. It's called the Langseth. This is a picture of it here. And this expedition that we did to Station Aloha was about a 3-week research cruise. Oceanographers like to name their expeditions, and my team calls this the Mahalo expedition. Mahalo means thank you in Hawaiian. And we were saying thank you because this expedition was funded by the Simons Foundation, and they actually paid for us to take 2 ships to the same spot, which never happens. And that's how we were able to get all of these people to this site for 3 weeks to take all of the measurements that we needed for this experiment. The ship I was on is the RV Kilo Moana, which is operated by the University of Hawaii. And so we ship all of our gear out there. We set up laboratories on the ship. It takes days to load and set everything up. We have to tie everything down so that it doesn't fly off the bench when the ship starts moving. And I just wanted to show you one more picture of the ships that we go on, again, for these long research trips, because it's really special to have a research fleet. You might see it here. It's that one. It's always a little demoralizing loading the research ship right next to a cruise ship because you think, I'm going to be on this one for 4 weeks. I don't think you guys are. So what we did is we did an experiment where we put out a drifter in this red patch of water north of Station Aloha, which is a feature in the physics called an eddy. That's just trapping a water mass in a circulation pattern. And we did this, and we put the drifter there to try to track the same population over time as it was moving around. And then we took samples every 4 hours for 6 days, nonstop, twice. And we did this because we were trying to use the light from the sun, which we know provides the energy for that daily flux of carbon into the system, as a driver for saying, okay, every day there should be this new pulse of photosynthetically fixed carbon going through the system. All right, so this meant being out there on the ship, taking samples, and filtering lots of water to capture the cells for our molecular analyses, and doing this daylight and at night in the dark and with red lights, because I was very worried we were going to light contaminate our samples, and the signals that we're looking for turn over very quickly and can change within minutes. I was on this cruise. You can see me looking a little tired in the background there, but this was a really exciting opportunity to go in and measure RNA and biochemicals. I'm just talking about the RNA now. trace that microbial carbon through the network. So again, we're looking at RNA, which tells us about the activities. We're filtering all of this water, and then we sequence it. And each of these samples, we sequenced 100 million stretches of RNA. That was not even possible 5 years ago. These numbers are incredible. My whole thesis I did on one sequence of RNA, right? So these tools are amazing, but they also then are a big data puzzle, right? Because all you get back from those sequences are A's and T's and G's, and then we have to put 100 million pieces back together and say, who did you come from and what are you trying to tell us? And I have to say that in the middle of the night, in the red lights, trying to take these samples, I really had this major crisis on the screws. I had these students, their whole dissertations were writing on this project. We couldn't tell if we were getting enough biomass to get a signal. And I really thought, this is possibly the worst idea I've ever had. Like, what was I thinking? This is like at the very edge of what is like tractable. And so when we got back to the lab and we actually had enough material to even generate the sequences, that was relief number one. But then I sat there thinking like, oh, day, night, like will we really see patterns in the RNA that go up and down with day and night? And so can we really trace carbon metabolism this way? So first, we just said, okay, let's try to make it really simple, and let's go back to our old friend, EHUX, our coccolithophore, which should be out there. We have a genome, which makes it a little bit easier to put that puzzle together. And we said, do we see any day-night patterns in these carbon pathways for EHUX? Look at that, right? So what you're seeing is the relative signal of RNA patterns in this one species from the environment. All of those gray blocks are nighttime signals. The white ones are the daytime signals. And what you see in the blue are all the pathways that make the pigments for light harvesting. In red encodes all of the enzymes and proteins that actually take the CO2 and turn it into the organic carbon. And you can see even before the sun comes up, that cell knows what's coming and it starts making all of its pigments and all of that machinery so that as second the sun comes up, it is ready to go and it starts fixing carbon. In the brown pathway, though, that's on the back end. That's the TCA cycle. So that's taking the organic carbon and then using it as a building block for growth and metabolism in the cell. So if you squint and think about it, you're basically seeing carbon go into the cell and start to underpin growth of that cell. This is work done by Maria Hernandez Limon when she was a Bridge to PhD fellow in my lab through Columbia's Bridge to PhD program. And I have to say, she did this work in my lab when she was a fellow. She then went on to a PhD program, and she just defended this year her PhD at the University of Chicago. So a whole new oceanographer out there ready to go. And we were so thrilled to be able to see these signals. And then, of course, what we did is start reconstructing them for the whole community. And I'll just briefly highlight the whole community responses here. So in this top pattern, again, the gray is night and the white is daylight. We see the POC, or the particulate organic carbon. So what you're doing here is seeing biomass grow up and then decline every day with that carbon that's locked up in cells. That carbon, when it decreases, means the cells are either getting eaten and moving to a different size fraction, or they're releasing that carbon as they die and break apart. So then we wanted to look at the same pathways now for the whole community that I just showed you for EHUX. And I've drawn over them here so you can see them better. But in the hot pink, you can see for the whole community, we see exactly the same thing. Photosynthesis starts getting, all those pigments start getting made. Then you see the green line moving to the TCA cycle. So we have photosynthesis and carbon fixation in the pink. And then as the cells are growing, you see how that green line overlays with the one above it, the POC, they're happening at the same time. So you can see how those metabolic processes, again, are moving carbon through different fractions in the system. And in fact, we can even see the handoff from the phytoplankton in green to those red and blue bacteria. They're shown in the yellow line here, and what you see here are sugar transporters, right? So what's happened is that the phytoplankton have made sugar. That sugar has been released to the environment, and right in the sink, those microbes, the red and blue bacteria, they don't know that the sun is out or not. They have no way to know, but they know that there's sugar coming, and they turn on those transporters to take that in and use it for food. So what you're seeing is the flow of that carbon through those different fractions of the community. And these new tracers of carbon flow really highlight the tempo and fate of carbon through that network. So there have been new opportunities now to move this work forward. In fact, we're expanding to doing some of these experiments in the Atlantic. And this is supported by a new NSF Science and Technology Center that we call Center for the Chemical Currencies of a Microbial Planet. Its acronym is CCOMP, and it's actually a UN Ocean Decade Science for Sustainability endorsed activity. And that brings me to the last point I want to briefly make, because I'm running a little long here, and that is that I think it's important to share this science with the broader community. So thank you for coming and listening today. But I've also been working to share this and what we're learning about the nexus between this network and carbon and climate with policymakers. And I was first able to do this at the UN Climate Change Conference, which we call COP, Conference of Parties. This is the meeting series that came up with the Paris Agreement, and this is where the decarbonization targets and emission targets are negotiated every year. It took, unfortunately, until COP27 for the ocean to be represented as both a player and a stakeholder in what happens next for our planet. But I had the privilege of joining the Ocean Pavilion team to talk about marine microbes at COP27 and try to advance their understanding. Then a year later, we had a whole special issue of Nature Microbiology, including a feature article that said, microbiology must be represented at climate talks. So this was great. And if we fast forward to COP30, which was last month in Belém, Brazil, I was there. I was really inspired I'm not going to take the time to read this quote, but even the Secretary General was talking about the importance of the ocean and ocean life and protecting it for our future. I talked about microbes at COP30, and we had a whole panel focused on advocating for this invisible majority that's so important for our planet. And I'll just wind down by saying, even last Friday, on Science Friday, there was a piece called "Can a Microbe Conservation Movement Take Off?" So I feel like maybe the time has come where microbes and ocean microbes, from my perspective, are starting to gain traction in terms of their importance. The time is now. There is a confluence of need and opportunity to more fully resolve the rules that govern this microbe chemical network and its role in ocean health and climate. And collectively, this knowledge will help us better predict ecosystem resilience on a changing planet. So I want to leave you with the thought that I have hopefully convinced you that microbes are amazing, and take questions. Thank you so much.